Hello student, good afternoon. Today we will start the main topic, that is the multiple alleles. In this topic, one article, that is the ABO blood groups. ABO blood groups. In the blood group system, there are 4 types of blood groups as A, B, AB and O. The A, B and AB blood group were discovered Carl Landsteiner in 1900, whereas O type was discovered by D. Castillo and Sterling in 1902. Blood type A, RBC with A antigen. Blood type B, RBC with B antigen. Blood type AB, antigen AB in RBC. And blood type O, no antigen in RBC of the O blood group. 4 blood groups of the ABO system with their antigens, RBCs and antibodies in the plasma. O blood group, no antigens in the RBCs. One table is there, the ABO blood group and their compatibility. Number 1 group, blood group A, blood cell antigen A, plasma serum anti B, small b, capital B, can donate blood to. A blood group can donate A and AB blood group person. Can receive blood from A blood group person, can receive O and A blood group person. Genotypes IA or IA small I. Second number, B blood group, antigen B, plasma serum anti A, can donate blood to B and AB and can receive blood from B blood group person, can receive blood from O and B. Genotypes IB or IBI. Third number, IB, AB blood group, A, B and AB. Third number, AB, antigens A and B, plasma serum none, can donate blood to AB, can receive blood from all universal recipient. AB blood group person can universal recipient A, B, AB and O. All types of blood can receive. And genotypes IA, IB. Fourth number, O blood group, blood cell antigen none, plasma serum anti A, B, can donate blood to all universal donor. All types of blood group persons can donate the O blood group and can receive blood from only O blood group. O blood group person can receive blood only O blood group. Genotypes IT. Group A, donors O and A, receive O and A, donate A, B and A. Group B, receive blood group O and B, can donate A, B and B. A, B and B. Blood group AB, universal acceptors, cannot be donated. Group O, donate AB. O blood group, universal donor. All types of blood group persons can be donated. Recipient only O blood group showing their compatibility. Landsteiner found that there were two different types of A and B antigens in the RBCs. He found that the cell can contain antigen A, B, AB or neither the both. Due to this situation, there are four types of blood groups. He also found that the blood serum always contained antibodies which would react with the antigens not present in the erythrocytes. For example, if a person had the A antigen but not the B antigen, he would carry the B antigen. He would carry the antibodies which would react with B anti B or B. A person with neither A nor B antigen, type O, would carry both anti-A and anti-B. Importance of blood transfusion. Blood typing is performed by mixing an individual's RBCs with appropriate antisera on a slide and seeing if agglutination occurs. Dangerous hemocytic transfusion reaction occurs when blood is transfused into an individual within an incompatible blood type, that is, an individual who has agglutinized against the red cells in the transfusion. As a result, donors' red cells agglutinate and hemolyze in the recipient's blood. Free hemoglobin is liberated into the plasma. The severity of the resulting transfusion reaction may vary from an asymptomatic minor rise in the plasma bilirubin level to be severe jaundice and renal tubular damage with the anuria and death. A transfusion can be given from a donor to a recipient of the same blood type without any complications because there will be no antibodies present in the plasma of the recipient which could cause agglutination of the cell being transfused. If incompatible blood is transfused, clumping always takes place of the donor blood. For example, if type A blood were given to a type O person, the anti-A abundantly present in the plasma of the recipient would clump the type A cells of the blood being transfused. On the other hand, type O blood can be given to person having A and B or AB type because antibodies present in the donor plasma active against antigens in the recipient circulation are rapidly diluted in the recipient's blood Summarize the possibility blood transfusion between various blood type. Group AB blood contains antigen AB in the cells but neither anti-A nor anti-B antibodies in the serum. Such serum therefore will not cause clumping of cells of any blood type. Persons having groups AB can receive blood from any other groups, hence called universal recipient. On the contrary, group O blood contains both serum antibodies but not cellular antigens. Therefore, such cells will not clump when mixed with serum of any blood type. Persons having group O therefore can give blood to any other group, hence called universal donor. Rh factor. The Rh factor was discovered by Landsteiner and Wiener from rabbits immunized with the blood of a macaque rhesus monkey. Macaque rhesus monkey, known as brown monkey. As we know very well that antigen is present in the RBCs, erythrocytes, and antibody is present in the blood plasma. RBCs contains antigen and that antigen is known as Rh antigen. A person having blood groups A shows presence of antigen A, RBCs and antibodies B in plasma. When the antigen is present, Rh positive name was given, and in some cases of monkeys, RBCs were without antigen and so were named as Rh positive. In Rh positive, there is no natural antibody in the plasma. Blood from rhesus monkey injected into rat and rabbits or guinea pigs produce antibodies D anti-D, which shows agglutination of RBCs of all rhesus monkey in the blood plasma. Anti-D is produced, which is the artificial antibodies. If corpuscles of monkey are injected to the human, then anti-D is developed, that is artificially antibodies is developed in the human being. Blood plus anti-D, rat plus rabbit plus man, 85% population given the positive reactions RBCs are H positive. Blood plus anti-D rat plus rabbit plus man, 15% population gives no reactions. Negative reaction RBCs are Rh positive, negative. Injected of anti-D are rat plus rabbit is known as sensitization. Antigen and antibodies are thus specific for the specific individuals. Genetics blood groups. Gene D is responsible for antigen Rh which present in the autosomes Rh positive. T is the dominant and small d is capital D is dominant and small d is the recessive. When Rh positive male marries with Rh negative female, it shows the following cross: Rh positive father, Rh negative mother. Capital D, small d, F1 generation, capital D, small d, all heterozygous Rh positive offspring. If such offspring as a interbreeds for the F2 generation, Rh positive, D, capital D, small d, and Rh positive, small d. F1 generation, capital D, small d, small d, small d. In this cross, 50% Rh positive and 50% Rh negative. Rh negative persons. Rh positive gives rise to Rh negative, sensitizes plasma, used to Rh antibody is developed and a serious reaction is there which shows clumping of bloods. The earlier transfusion is not dangerous, but later transfusion is dangerous as the antibodies development is continuously going on. Erythroblastosis. Rh factor is of great significance in childbirth. It has been found that an Rh negative woman married with Rh positive man in the pregnancy, when placenta is formed, it don't allow antibodies to enter in the intrauterine wall. Normal growth of embryo occurring up to 9 months and 9 days. At the parturition, the mixing of blood takes place. Blood of offspring and mother mixes. Embryo is Rh positive and mother is Rh negative. At such instance, the first delivery is normal, but the embryo has given the antibodies in the blood of the mother. Antibodies in the blood of the mother. Embryo antigen, placenta Rh, mother Rh negative. Embryonic contingence RBCs, hemolysis, embryo, migration of the antibodies in the embryo placenta for prevalence of Rh positive mother. In the second time, when the mother Rh negative is pregnant, the antibodies in the bloodstream of mother pass through the placenta and cause damage to the RBCs of the fetus. This causes the disease known as erythroblastosis feticis, which is characterized by anemia due to the hemolysis breakdown of RBCs in the fetus and consequent jaundices as the blood vessels in liver becomes clogged with the broken cells and bile is absorbed by the blood. The disease may be so severe as to cause death of fetus before birth or death within a few days after birth, that is, neonatal death may be occurred. Rogam method or preventing erythroblastosis fetalis. Rh positive negative mother is given to special blood test after delivery of her Rh positive child. If fetal Rh positive cells are present in the mother's blood, she is given injections of Rogam. Rogam is a preparation of negative Rh antibodies. It is obtained from immunized donor. The Rogam forms a coat around fetus RBCs in the mother's blood. Is result no Rh positive antigens are available to stimulate mother's circulation and no antibodies are formed, so normal delivery possible. The cost of this injection ranges from the 4,000 to 6,000. This is the ABO blood group. Thank you.